Good afternoon. My name is Dealwis. I'm from Case Western Reserve University. I'm going to talk about the cancer target ribonucleotide reductase and its allosteric regulation. I'm going to give you a brief introduction, especially about stressing on its allosteric regulation. And then I'm going to talk to you about the cancer target, how it's been targeted in the past, and how new future therapies are occurring, including my own efforts from our lab. This is an acknowledgement slide. People who have worked in my group in the past and present, including those who are collaborators from various parts of the world at different universities, and all of these people made major contributions to the project that I'm going to talk to you about. And I'm also acknowledging all the funding sources. So what is Rnr? It is involved in building the building blocks of life. It makes deoxynucleotide triphosphates. Its slowest step, where it turns RNA molecules into DNA, and hence it's a major cancer target. And here the reaction is shown where the 2' hydroxyl on the sugar is reduced to make from RNA to make DNA molecules. It sits in the transition between the RNA and DNA worlds. So just like reverse transcriptase, sophisticated mechanisms through DNA occur because of this enzyme. The other major important role that Rnr makes is maintains a balanced nucleotide pool. I'm showing here a typical distribution of the 4 nucleotide triphosphates in a Drosophila genome. And as you see, it's asymmetric, they're not all equal. But if you skew this nucleotide pool a little bit either way, whether it's to make one of the nucleotides greater or lower, this ends up in causing mutator phenotypes that can lead to cancer. So it's very important that this enzyme has to regulate this nucleotide pool in the body so that life can be maintained. Now, Rnr involves 2 subunits. There is a catalytic subunit called alpha, and then there is a beta subunit that is housing a free radical that requires to be transferred to the catalytic subunit to start the reaction. This happens through a very elegant chemical mechanism called proton-coupled electron transfer, where this catalytic radical has to move from the small subunit into the large subunit through a shuffling process, traveling some 35 angstroms to initiate catalysis, where the ribose receives the free radical. Now, what I'm showing here is the minimal active form. In one of the figures is alpha 2 beta 2, where an alpha 2 dimer and a beta 2 dimer have to come together to form the minimal catalytically active unit. But in mammalian RNAs, such as human, this is more complex. It forms an alpha 6 hexamer, where the catalytic subunit forms a hexameric form, and it can couple with alpha 2 beta 2, alpha 2 beta 4, alpha 2 beta 6 active forms, or an alpha 2 beta 2 inactive form. Now, I'm showing you the large catalytic subunit where the reaction happens. This is where a lot of the action happens. There are 2 allosteric sites where there is an A site. The activity is regulated by ATP activating the enzyme or DATP binding at the A site that inactivates the enzyme. So you have allosteric activator and inhibitor. And then there is an S site where allosteric effectors, 4 types of allosteric effectors will bind. And depending on which binds, it selects for 4 possible substrates, which I will show you a little later. Now, how does Rnr maintain a balanced nucleotide pool? At the molecular level, our lab with others, demonstrated this process where you see two major loops. One of them is involved in effector binding, that's called loop 1, where a catalytic effector such as ATP or DATP, DGTP, and such can bind, and then it'll make a loop 2, which is usually in a close position in the active site, which cannot bind substrates because it is sterically blocked, will be moved in a favorable way to complementarily choose the appropriate substrate. And this is how this amazing allosteric regulation happens to select for 4 possible substrates to produce the 4 products. Now here, we're showing the 4 possible complexes that we originally determined with the yeast enzyme, and then we have also subsequently showed this with the human enzyme. Now, so the 4 possible substrates it could be ADP, GDP, CDP, and UDP. And what happens at the beginning of the cell, the cell, as it enters this S phase of the cell cycle, where proteins have to be replicated for replication processes, there's ATP at about a 5 millimolar constant concentration in the cell that will bind at the A site, activating the enzyme, but also bind at the S site, and it selects for pyrimidine substrates, CDP, UDP, and then later TTP, as TTP gets upregulated, it'll bind and select for GDP, and DATP will select for, DGTP will select for ADP, and as ADP becomes DATP through phosphorylation processes, it'll bind at the activity site and then self-regulate it by shutting down, allosterically inhibiting the system until the DATP levels are used in the cell, and then the whole system is activated. This happens for 8 hours through the S phase of the cell cycle. Now, how does the regulation happen through ATP and DATP? We were able to show that ATP binds at the ATP binding site and DATP binds at the DATP binding site. We solved the structures using X-ray crystallography, which is shown here on this slide. And what you see is that ATP has a much lower affinity for this site. It has to, otherwise the enzyme would be dead. DATP has like 1,000-fold improved affinity for the same site. And the way the enzyme achieves this is by the extra, the deoxy form, which is DATP, is able to bind deeper in the pocket, make more interactions, and therefore it gains greater affinity. Whereas ATP binds more shallow in the pocket, and therefore it's able to activate the enzyme, and it's not inhibiting. But as I will show you, there's a requirement of hexamerization for inhibition that is required with the allosteric regulation of this protein. Now, this shows oligomerization regulating RR. We were able to determine the first structure of the alpha-6 beta-2, both with X-ray crystallography and cryo-EM. Now, what happens here is we were able to show that the inactivity of DATP is achieved by making the beta subunit bind within the alpha subunit or close to the alpha subunit further away so that the free radical cannot be productively transferred to the catalytic site, and therefore the enzyme is inactive. But what this diagram is showing is through using multi-angle light scattering, we show that as the DATP concentrations change, the alpha goes from alpha to alpha 2, and then alpha 2 becomes alpha 2 beta 2, or it can become an alpha 2 beta 4 potential complex, although we have not visualized it. But eventually it ends up as an alpha 6 beta 2 inactive DATP complex. Now with ATP, the story is different. It can probably form alpha 2 beta 2, alpha 2 beta 4, alpha 2 beta 6 complexes that are active. So the key is, how does ATP, which is also a hexamer, and DATP, which also induces a hexamer, able to form an inactive versus an active complex? We cannot structurally fully answer this question yet because we don't have an ATP hexamer structure. But through mutagenesis, we have shown that there are structural differences that may account for this phenomenon. Now I'm going to switch my talk to cancer and how Rnr is inhibited by cancer drugs. So there are two types of inhibitors, two classes. One target the large subunit, and these are mainly nucleoside, but I'm also going to talk about some of our effort that are non-nucleosidic. And these nucleoside drugs, which I will show later, are used as chemotherapies in the clinic. And then there is, on the beta subunit, there is one drug that's approved, hydroxyurea, which quenches the free radical. So these are the kind of drugs that are found. Now this table shows you all the anti-metabolite class of chemotherapy agents. I want you to pay attention to the Rnr drugs that are like clofarabine, cladribine, gemcitabine, fludarabine. These are all nucleoside drugs, and they target the large subunit. Now what's interesting is gemcitabine is a very unique drug in this class, because gemcitabine is the only known drug that can penetrate solid tumors. So it is used just about with all chemotherapeutic regimes against things like breast cancer. It was approved against pancreatic cancer. It's one of the few known drugs approved against pancreatic cancer. Even though it's a poor drug, it only improves life by about 3 months. But nevertheless, like I said, and the other drugs that target the ribonucleotide reductase, they are not good at penetrating solid tumors. Part of the reason for this is there is dose-dependent toxicity. They cause myelosuppression. The neutrophils and cytopenia effects, they go up with these drugs. But gemcitabine can be used within its therapeutic window to deal with some of these cancers. And the way that gemcitabine works is that it actually binds the catalytic site of the large subunit. And we have solved the structure showing how it binds, which I will show later. And it inactivates it in an irreversible manner. So it covalently modifies, kills the enzyme. But its main form of toxicity, as with all the other nucleosides, is through chain termination. Now, as far as the other nucleoside drugs go, they are DATP analogs. They basically form inactive hexamers and inhibit the enzyme that way. Now, why do more screening and why bother making more Rnr drugs? As I mentioned before, they are very toxic, the ones that are involved in. They're very polypharmacological. They have many, many targets. So we're looking for something that is a small molecule that will be reversible, not like gemcitabine, which is irreversible, but will have greater safety profiles and have more efficacy advantages of therapeutic advantages, and this is what we're looking for. So that's the reason why we wanted to screen. So what we started is we're using an approach called rational drug design or knowledge-based drug design, where we take the structure of the protein, and because we have solved the human structure, and we're able to screen the catalytic site or the allosteric sites computationally in silico by screening hundreds of thousands of compounds. This is the strategy we are using. We are screening using a program called Schrödinger. We take a library of 350,000 compounds. Then we validate the hits experimentally using fluorescence quenching. If we get at least 20% quenching, we think that these are good hits. Then we do enzymology to find the mode of inhibition. And finally, we will also calculate IC50s by experimentally determining the growth inhibition in cancer cell lines. So that's basically our strategy. And this is showing a hit compound called NSAH. This is an N-salicylic acid hydrazone molecule. And what we show here is the fluorescence quenching. As we add the compound, you see quenching, and it is showing binding to the enzyme. And then we were able to solve the structure of NSAH bound to the target human ribonucleotide reductase. And what we show is it binds in the catalytic site. And then we are also showing that it's binding within the active site, same place as its substrate GDP. And then finally, we show how it's interacting with the active site in the last panel. So, as I said, we solved the structure of NSAH, but we also solved the structure earlier on with gemcitabine, and it is showing that it is binding at the same site as gemcitabine, the cancer target. And then below that, I'm showing some of our docking studies, as well as theoretical pharmacological properties. For example, the drug satisfies the rule of 5, Lipinski rules, and it is membrane permeable, and it has a reasonably good log P, which would make it a reasonable oral drug at this level. Okay. Now this is showing how the NSAH is acting upon the active site, and is showing you that it binds roughly around where the GDP, but it's doing chemical mimicry. So, for example, it's showing you that the naphthyl group is binding where the phosphate is roughly binding, and it is showing that the hydrazone is binding where the ribose molecule is binding. And then it is also showing you that the base would be occupied by the phenyl ring, the hydroxyphenyl ring. So it's not binding exactly the same way, but it's trying to chemically mimic it, and that's what you expect a drug to do. Okay. Now, here we're trying to compare how good this drug is against gemcitabine. And what I'm showing you here is we're looking at bone marrow cells. This is an assay done with bone marrow cells. The reason why we choose bone marrow cells is because gemcitabine is extremely toxic to bone marrow cells, and that's why it's not a great therapeutic. So what we do is we show here that gemcitabine in a colony forming assay is killing all the colonies at around nanomolar levels, whereas NSAH is 1,000-fold safer, and it's hardly killed any cells at the micromolar level. So we expect that NSAH to be a safer therapeutic for treating patients. Okay, now another way we confirm that it is hitting the target in the cells is we can measure the nucleotide triphosphate levels, the 4 nucleotide triphosphates. And if it is hitting RNR, then it should deplete the deoxypurine pools, DATP and DGTP. And we also do control experiments with typical RNR inhibitors, hydroxyurea, as well as gemcitabine. And the trend appears to be very similar. It is depleting the DGTP and DATP pools. So we are fairly confident that NSAH at the cellular level is an RNR inhibitor. Now, we have done in vivo studies, which I haven't shown too much here, but in generally, they are inhibiting growth inhibition in mouse models, in xenograft models by about 70%. They don't lose any weight. They seem to be happy mice. And so this would suggest that NSAID is a low toxic molecule. Now, I haven't talked beyond this, but we have done several rounds of medicinal chemistry, and our latest drug, so NSAID was our original lead compound. We have moved to next generation drugs, which I'm not going to talk in this talk, that are looking very promising. This is a summary of my talk. Briefly, I gave an introduction to ribonucleotide reductase and our contributions given to the field. And finally, I start to talk about, in the latter stage of my talk, about the cancer target ribonucleotide reductase. I talked about the existing drugs, their downfalls, and what new strategies are used in my lab to overcome these problems. And I'd like to thank you for your attention.